Hi, I'm Joy Fan, a 7th period AP Bio, and I'm with Ben X, the science guy, on cellular respiration. The joys of exerting your pent-up energy on metallic objects. It was a beautiful and sunny day when Ben and his buddies decided to work out together. And they did, for a while, until they became exhausted. Isaac, do you feel tired?
Yeah.
So they decided to take a food break. Immediately, they felt more energized. It was the wondrous phenomenon of cellular respiration. Brought to you by Hot and Spicy Noodles. Everyone loves the burn. You might have noticed that you can get really tired from working out. This is because your body is trying to produce enough energy so that you can do work. This all starts in the mitochondria, which is a membrane-bound organelle found in almost all human body cells. Its function is to perform cellular respiration and produce ATP. Now let's go to the lab. The main purpose of cellular respiration is to create ATP. So in the equation, we need ADP and inorganic phosphates. We need 30 to 32 of each. We'll also need glucose, as it gives us the energy we need to produce ATP. And we'll also need 6 molecular oxygen molecules so that we can use it to power the electron transport chain. For the products, we start with 6 carbon dioxide molecules and 6 water molecules. And most importantly, we have 30 to 32 ATPs that we can use in our body to do work. Try this. If you're ever feeling tired or unmotivated to exercise, make yourself a bowl of hot and spicy noodles. Today, I'm here with your favorite science guy, Ben. He's eating hot and spicy noodles at his house. Ben, how do you feel? I feel so happy I could eat another bowl. 15 to 30 minutes after Ben finishes his meal, his blood sugar will increase because of the increase in glucose. He should feel more energized and he's ready for cellular respiration. After Ben finishes his noodles, he needs to break them down. Noodles are made of flour, which is made from wheat and other grains. Flour is also very high in starch. Starches are polysaccharides that can be broken down into glucose by enzymes. Glucose is the monomer of starches and can travel freely through our bloodstream as it's polar. This is why Ben's blood sugar level increased. Glucose can enter cells through facilitated diffusion because it's too big and too polar to go on its own. But for the mitochondrion specifically, glucose must be broken down into pyruvate first. Another important reaction to cellular respiration is molecular oxygen. The air sacs in our lungs fill with molecular oxygen, which is taken by the capillaries into our bloodstream. There, the molecular oxygen can travel throughout our body and can go into cells through passive diffusion as a small nonpolar. Consider the following. Imagine you're working out with Ben and his buddies. When you lift a weight or play ping pong, you're using your muscles. The muscle cells need a lot of ATP to be able to function and move. Ben can also use his muscles to make more hot and spicy noodles so that he can produce more ATP. Humans are multicellular eukaryotic organisms, and compared to prokaryotes, we're much more efficient because we have mitochondria. Our mitochondria has 2 selectively permeable membranes that only allow certain things to go through. We also have the outer compartment and the matrix that are small containers so that the bumpage of enzymes and substrates increases. Also, the inner membrane made of cristae are little humps and folds that increase the surface area and allows more enzymes to be there. The cell is membrane bound and it contains many organelles in the cytosol. These organelles are bouncing around and this is the cytoplasm. In the cytoplasm, there's also the nucleus that contains the DNA and genes that are manuscripts for making things like enzymes. In the cytoplasm, there's also mitochondria that perform cellular respiration and make ATP. This is the mitochondrion. It's a double membrane-bound organelle with an outer membrane that has porin or holes. The inner membrane has cristae, which are little humps and folds that give it more surface area and allow more enzymes to be present. Therefore, the mitochondria is very efficient. Along the inner membrane, there's the pyruvate entry complex where pyruvate enters. And inside is the matrix where the Krebs cycle happens. And also along the inner membrane, there's the electron transport chain and ATP synthesis. Now we can finally start cellular respiration. The first process is glycolysis, which happens in the cytoplasm. In the cytoplasm, there's glucose and two enzymes floating around, kinase and dehydrogenase. Glucose is broken down by these two enzymes into two pyruvates. This is because glucose itself is too big and too polar to be able to enter the mitochondria on its own. Once inside, the two pyruvates give enough energy to the mitochondria to be able to produce ATP. It's important to note that glycolysis is an anaerobic process so that it does not require oxygen. In the energy in phase of glycolysis, glucose gains an inorganic phosphate from hexokinase and ATP, and another inorganic phosphate from phosphofructokinase and another ATP. Then this is broken down into two parts, which are two 3-carbon things or two phosphoglyceraldehydes. Here, two ATPs are used. Now, in the energy out phase, these two are used as substrates for our special dehydrogenase. The dehydrogenase has an NAD+ and an inorganic phosphate. The NAD+ gains 2 electrons and a proton from here once the 3-carbon thing is phosphorylated. So we have NADH here, and we have the 3-carbon thing, which now has 2 phosphate groups. Then this moves on to 2 kinases, where ATP will be made. So on the kinase, there's an ADP, and it takes one of the phosphate groups from our 3-carbon thing, and then it smushes the ADP and phosphate group together to create ATP. And the second kinase does the same thing with the phosphate group and smushes it together with the ADP to create ATP. And since we have 2 3-carbon things, that would be 4 ATPs in total. But since we have already used 2 ATPs, the net gain is only 2 ATPs. Also, I wanted to add that this is substrate level phosphorylation because the kinase just pushes the ADP and phosphate groups together. It does not require oxygen, so it is anaerobic. Now we're on to the pyruvate entry complex. There are pyruvates that are entering from all around the inner membrane. So for this, we start with 2 pyruvates that first meet a decarboxylase. So this is our pyruvate, and when it meets the decarboxylase, this carboxyl group rips off and becomes carbon dioxide. Then this 2-carbon thing meets up with a dehydrogenase, and this covalent bond comes off. And the covalent bond, the 2 electrons, go to NAD+, and with another proton, it becomes NADH. And finally, the 2-carbon thing meets up with a coenzyme A smooshase that adds A, and this becomes acetyl-CoA. So we have carbon dioxide, NADH, and acetyl-CoAs coming in from all directions into the matrix. In total, we have no ATP. Next in the Krebs cycle, the products along with a bunch of different enzymes are bouncing around in the matrix. This is really efficient for Krebs since the matrix is a small container, so the bumpage of these things increases.
Isaac, give us a beat.
Oxaloacetic acid is the first substrate, taking 2 carbons from the product of pyruvate.
That's acetyl-CoA. It starts Krebs. It makes a citric acid,
which is the select. This process uses 3 main enzymes. They make the reaction EPC and are used all the time. First is dehydrogenase, which gives us NAD+. It becomes NADH and carries electrons like a school bus. Then is decarboxylase, which makes CO2. Removes carboxyl groups and throws them out of the crew. Finally, there's kinase, which makes ATP. ATP is phosphorylated through the substrate. You see, in the very end, there are
2 ATP strings.
The other products found
around but are not relocated.
Not relocated!
Ben has 4 pet rock enzymes in movie theater butter, and they love to throw protons across the inner membrane. Pet rocks 1, 3, and 5 are the only ones strong enough to throw protons. They use the power of redox reactions to throw the protons, and electronegativity powers the entire electron transport chain. Each rock enzyme is more electronegative than the last, so they pull electrons from their friend on the left. And at the very end of the chain, there are oxygen molecules that are the most electronegative. They keep the electrons moving as oxygen wants to bond with the electrons to create water. At the beginning of the electron transport chain, there are two electron shuttles, NADH and FADH2. When NADH is there, it breaks off into NAD+, a proton, and two electrons that go to our first enzyme, the fruit rock. Once this happens, the fruit rock gains energy from the redox reaction, changes shape, and pumps 4 protons across the membrane. After, the 2 electrons go to the fat, as it's more electronegative than the fruit rock. The fat doesn't pump anything but gives the 2 electrons to APA, who changes shape and pumps 4 protons across the membrane. Then the 2 protons go on to the cute ladybug, as it's more electronegative. This one doesn't pump anything but gives the 2 electrons to Pikachu. Pikachu changes shape and pumps 2 protons across the membrane. In total, 10 protons were pumped. When FADH2 is used, it becomes FAD, a proton and 2 electrons. FADH2 is more electronegative than our first enzyme, so it must give its electrons to the fat. The electrons travel through the chain where 4 protons are pumped through APA and 2 are pumped through Pikachu. In total, FADH2 pumped 6 protons. Did you know that the shape of ATP synthase is very similar to that of a lollipop? And since we need ATP to be able to live and function, the enzyme is the lollipop of life. The inner membrane doesn't let protons through. That's where LOL comes in like a train that goes choo-choo. It lets protons through with facilitated diffusion. It's called chemiosmosis and it's part of the profusion. Protons are abundant in the outer compartment. They can't pass through the nonpolar department. They want to move from high to low concentration. ATP synthase helps with that and energy formation. The lollipop is spinning when protons enter. It makes ATP all around and in the center. This turns potential energy into a kinetic one. So the more protons there are, the more fun. 4 protons are needed to make 1 ATP. This is powered by oxygen, you see. As O2 is super electronegative and pulls electrons through, this makes the gradient and LOL runs really smooth. This is called oxidative phosphorylation, and it makes ATP. It's really quite nifty, don't you agree? The O2 accepts electrons, but it needs to be stable, so protons from the matrix are attracted like a chair and a table. H2O is made and another oxygen takes its place. This continues until no more oxygen is left in the space. You can think of the lollipop of life like hydroelectric dams. The water from the reservoir goes through the turbine, generating energy, just like how protons go through the ATP synthase, makes the LOL turn, and that creates ATP. A gradient is the difference in concentration of something across a membrane. A gradient is potential energy as things like protons want to move from high to low concentration. An analogy would be these cows here that are crowded behind this fence. They would love to move, and their potential energy becomes kinetic when they're able to move. This is like cellular respiration, as protons can move across the membrane once there's ATP synthase. So far, we have a total of 2 ATPs from glycolysis and 2 ATPs from the Krebs cycle, which are through substrate level phosphorylation. In our matrix, we have 8 NADHs and 2 FADH2s that create a total of 23 ATPs, so far we have 27 ATPs. The rest come from the 2 special NADHs that are made through glycolysis and are found in the outer compartment. So the lazy Susans, you can either have the first one or the second one, but you cannot have both. So when the first lazy Susan is used, there are 5 ATPs created. When the second one is used, there are 3 ATPs created. This is why some cells make 30 ATPs and others make 32. Now that we have more ATP, we can use our muscle cells to exercise. When ATP goes through hydrolysis, intermediates can be phosphorylated to do work. After working out with her friends, Joy's arms are starting to feel tired. Joy's muscles are in need of more oxygen. If there are not enough oxygen molecules, the ETC will be backed up since there's nothing left to accept electrons. The only place where ATPs are still being made are in glycolysis because it's an anaerobic process. But fermentation is needed as there wouldn't be enough NAD+s. Lactic acid fermentation takes pyruvate and turns it into NAD+ and lactic acid. When Isaac goes through strenuous activity, he has to use lactic acid fermentation, which causes a buildup of lactic acid. This changes the pH of his muscle cells, so water comes in to try to neutralize it. When this happens, the muscle cells start to swell, and they push up against pain receptors, which is why Isaac is sore the next day. Fungus fermentation is when yeast uses pyruvate to create NAD+ and ethyl alcohol. When grapes are used, further glucose and pyruvate, wine is made. When corn is used, whiskey is made. And when molasses from sugarcane is used, rum is made. Well, that's our show, folks. Thank you for watching.
When it happens.
Yeah. How do I use when I'm in the middle of the game?
No, literally my teammates are like literal shit.
It doesn't matter, but I soloed. I just.
Bully.
Oh my god, that was so. I think I broke my rib cage.
What?
Do your muscles feel depleted?
No.
Oh.
One such component is the cytoplasm. The cytoplasm has many molecules, and just for clarification, I want to state that glycolysis has